Our immune system is a powerful biological machine made up of specific cells and organs that protect the body from infection and disease. Among these are T cells, which hunt down and destroy abnormal cells, including cancer cells. This process is highly sophisticated. T cells have protein receptors, or antennae, on their surface, which are designed to attach themselves to protein fragments found on the surface of all cells, called antigens. Sometimes these antigens can be abnormal, alerting the T cell to take action. When a cancer cell is discovered, T cells release killer chemicals to destroy the abnormal cell and recruit other T cells to support it in its elimination. Unfortunately, cancer sometimes develops ways to fool our immune system and evade its defenses. It does this by creating multiple abnormal antigens, which prevent our T cells from latching on and identifying abnormalities as they normally would. Or by disguising itself as healthy cells, meaning our T cells stop recognizing them as foreign and no longer do their job of destroying them. This is how cancer develops. To combat this, CAR T-cell therapy, a novel type of immunotherapy, is designed to strengthen and harness the immune system's inherent cancer-eliminating powers and remove its disguise. CAR T-cell therapy is the name given to chimeric antigen receptor, or CAR, genetically modified T cells. It is currently used for the treatment of blood cancers. Traditional immunotherapy usually attaches a drug to a patient's T cells, enabling them to see and destroy cancer. But CAR T-cell treatment is created from a patient's own T cells, engineered in a lab to be reinfused in large numbers to recognize and destroy cancer cells. There are no drugs involved. Firstly, thousands of a patient's own T cells are collected in a process similar to a blood donation. Blood is removed intravenously from the patient's arm. It then goes through a cell separating machine, which filters out the T cells and returns all other blood cells back to the patient. This process is called leukapheresis. Separated T cells are sent to the lab, where they are genetically altered by adding the specific chimeric antigen receptor, turning them into CAR T cells. These newly engineered cells are expanded in the lab, and millions of them are infused back into the patient following a short course of chemotherapy. Their new receptors enable them to latch on to a specific antigen on the patient's tumor cells and destroy them.